Welcome everyone. My name is Serap Vatansever. I am a postdoctoral research associate at the Department of Dairy and Food Science at South Dakota State University. Thank you for joining us for the effects of pH on the techno functionality and interfacial activity of potato protein extract, which is originally published as an e-poster at the 2021 AOCS annual meeting and expo. Today is the special edition AOCS annual meeting webinar is presented by Dr. Ali Jafarpour. Now I am going to introduce a little bit about the Dr. Ali Jafarpour. He's a senior researcher at the Bioactive Group Analysis and Application of National Food Institute at the Technical University of Denmark. He received his PhD from the MIT University in Melbourne, Australia in 2009 and started an academic career at the Surrey Agricultural Sciences and Natural Research University in Iran from 2009 and 2018. Dr. Jafarpour is his primary research interest is value addition techniques of the side streams from the different food sources with emphasis on the seafood waste with a focus on food biophysics, biochemistry, on protein gel networks in terms of protein peptide profiling, texture, rheology, and microstructure. The current core of his research is the techno-functionality and structural function relationship of enzymatically derived peptides from the plant and animal sources. Please write any questions for our presenter into the question section of your control panel. All questions will be addressed at the conclusion of the presentation. Thank you again for joining us. Now we can listen e-poster recorded by Dr. Jafarpour.
Hello, my name is Ali Jafarpour, and I'm a senior researcher at Technical University of Denmark, and here I'm presenting some results regarding techno-functionality of potato protein extract, which is actually a part of a bigger project known as PROVIDE, which will focus on valorization of different sorts of side streams by application of enzymatic hydrolysis, proteomics, and bioinformatics. So here in terms of potato, I would say that during the extraction of the starch from potato, a huge amount of wastewater is produced that is called potato fruit juice. PFJ contains considerable amount of different fractions like vitamin, minerals, and specifically proteins. And protein content of PFJ is between 1.5 to 4%. Currently, in the industry, they apply heat coagulation to recover this part of protein. But we know that this treatment protein loses its functionality. So we were curious to find out if you recover protein in an intact form, how would be its functionality. So I received actually the potato protein extract in a lyophilized form from the industry, which contains 87% intact potato proteins. And I made a protein solution of 0.5% and different pH, in the range of pH that start from 2 up to 10, and I measured different bulk functionalities, including solubility, emulsifying activity and stability, droplet size, zeta potential, and interfacial activity. So in terms of result, I start with the solubility, and as we see here, we had a very typical V shape of solubility in a range of pH that shows a very high solubility, more than 90% at the acidic region and more than 70% alkaline pH 5. We had the V shape or the drop of solubility at pH 6, which shows this type of protein is pH dependent. And in the next step, I measured the emulsifying activity and the stability by making emulsion containing 25% oil. So what we see here in a blue bar indicates emulsion activity index, and what we see a clear shift from acidic region toward the alkaline region, which shows increasing the emulsion activity at alkaline region. And in the orange line, it shows the emulsion stability. So the emulsion that we made in alkaline pH, rather than having emulsion, higher emulsion activity, shows the higher emulsion stability. Well, to justify these results, then we measured the droplet size and zeta potential, and the blue bar again shows the droplet size. So clearly at pH 2, what we see here is very high droplet size at acidic region, while by moving toward the alkaline region, the droplet size significantly reducing, while the zeta potential, which is indicated with the orange line here, increasing in terms of higher negative values, which shows more than minus 30 in terms of zeta potential. Finally, I measured the interfacial activity by making the potato protein droplet in oil as ambient phase. So, and we came up actually with very interesting results. In a pH from 2 up to 6, after making the droplet, actually the interfacial tension start to reduce, and passing the time, it's trying to reach to the near equilibrium. What happened during pH 8, which is indicated with the yellow line here? So what we see here after a couple of minutes, before 3 minutes, the droplet is detaching from the needle, and the same event happened for pH 10, which is indicated with the dark blue line here, which actually happened even before 1 minute after replacing the oil. So what would cause this phenomenon, still we don't know, but we can actually speculate that here the protein shows a very high surface activity at the alkaline region. Here to conclude the result, I would say in terms of solubility, the acidic pH causes higher solubility of the potato protein extract compared to the alkaline pH. In terms of emulsion activity and stability, emulsifying capacity of PPE is significantly high at alkaline range of pH compared to the acidic region. And in terms of zeta potential and droplet size, the PPE solution aligned, this result actually aligned well with the emulsifying properties. And finally, in terms of interfacial tension activity, the detachment of what we saw was detachment of the droplet at the alkaline pH, which can be attributed to the synergistic effect of high protein concentration of aqueous phase, which was 0.5%, which normally measure the interfacial activity, interfacial tension activity. The protein solution or protein concentration solution would be around 0.01%. And also the pH dependency of interfacial activity of potato protein extract that shows very high mobility and flexibility at alkaline pH, but still it needs to be clarified. Thank you very much.
Thank you for this e-poster recorded by Dr. Jafarpour. It was very nice to hear about it. And then, if you have any question, please provide your question at the question part of your control panel. Now, I also had several questions that I want to ask Dr. Jafarpour until we are getting some questions from audiences. It was very nice to hear about your research, Dr. Jafarpour. And then basically my first two questions are related with the material that you use for this research. And then it seems that you use a potato protein extract, which is as a powder form. And then my first question is that how did you obtain the potato protein extract from the byproducts of the potato processing? I mean, which procedure did you use to obtain this extract?
Yeah, first of all, thank you very much for inviting me to present my result in AOCS conference. It's a very interesting question, actually, because there are two different types of potato protein. I would call them one of them potato protein concentrate, which is the denatured protein, obtained by heat coagulation or acid precipitation. And the sample that we use in our experiment, I would call it potato protein extract. I didn't I didn't perform any specific experiment to obtain this material. I received this material from the company.
Okay, so commercial.
And yeah, exactly. And I wouldn't say anything here to just, it's come from industry and they don't want to keep their result confidential. But I assume as a scientist, we know how we can provide the protein in an intact form.
Okay, okay. The reason that I asked the question, because as I know, based on the wet isolation methodologies and the drying period, it directly impacts the functional characteristics. For that reason, I want to ask this question.
Yeah.
And also the other, when I read the introduction at the poster, I see that these potato protein extracts obtained from the potato protein fruit juice, I guess, potato fruit juice.
PFJ, yeah.
And when I see that around, I think 50% of this juice is inhibitory proteins. Another question was that, did you have any experiment about the content of the inhibitory proteins for this extract before conducting these functional experiments? It is another material-based question. If you have an answer, I would like to hear about it. If there is not, I would like to ask another question for the functionality.
Let me clarify your question. You're asking whether I inactivated the protease inhibitors, kinase protease inhibitors specifically or not?
Yes.
Yeah, no, I didn't, because I know in my previous experiment, I had some difficulties with this type of protease. So I would say to do the enzymatic hydrolysis, I didn't do anything to inactivate this type of protein. But our focus was on the patatin part, even though I know the kinase protease, including more than 50% of the protein. But no, I didn't do anything to inactivate that.
Okay, okay. Thank you. Thank you so much. It was very nice to hear. And yeah, I am checking the question panel. Yeah, I think we didn't have any from the audience, but I also have another question about the functionality that I want to ask. When we see your poster, basically you check the functional characteristics in terms of protein solubility and emulsion properties at the different pH level. And then basically you obtain the protein solubility almost significantly higher at the pH 2 compared to others, especially alkaline pH. But when we check the emulsion characteristics at pH 2 and 4, we really have the low level emulsion activity index and emulsion stability. My knowledge based on the proteins, mostly there is a positive correlation between protein solubility and emulsion properties. For that reason, I want to ask, it is not always correct that to get to this statement. And then for the potato protein, what is the exact reason why we are getting the very low level? I think the significantly low level emulsion activity and also stability indexes at pH 2 and 4, even though we have very high solubility. What is the main reason about it?
Yeah, this is true actually, and we were expecting to getting the higher emulsion, higher solubility at the alkaline side actually, because I have done some other experiments. Yeah. But we couldn't come up with a clear answer for the question, but this is the result that we got. I would say this was sort of preliminary experiments that I did on this sample. I didn't focus too much on these details because we were shifting to some other parts, and this was sort of warming up work for me. So I know, and it was interesting actually. This is why I present this data here in this conference, because we were expecting to get a higher solubility in alkaline side. But the possible explanation might be, I don't know, because I made a protein solution of 0.5% protein solution might be related to the concentration of protein. And also, as you mentioned, in our experiment, we didn't focus just on the pathogen part. We had different protein fraction, including the Kuhn's protease inhibitor. That might be some side effect of the protease inhibitor.
Okay. That is why I want to ask a question related to protein inhibitory proteins. I was just thinking might be it is the reason or not. For that reason, I asked the previous question related with the inhibitory proteins.
Yeah.
And
yeah, but thank you very much for asking very critical questions.
yeah, because I became really surprised to have that higher solubility at pH 2 in particular.
Yeah.
And yeah, those were my question overall for the study. I think when I checked the question, I think we didn't have any question from the audiences. So it was very nice to have this presentation together. I think we can right now conclude our webinar here. And if you have any additional things that you would like to share, so we are happy to hear from Dr. Jafarpour. I hope I am pronouncing your surname correctly. Yeah, it was very nice to have you.
Yeah, thank you very much. Yeah, you pronounced it correctly.
Thank you.
Thank you very much.